Hey, how's it going, everyone? This is FixerMed. Welcome back to my high-yield biochemistry review series for the USMLE Step 1, NBME CBSE, and NBME CAS examinations. This will be part 20 of a multi-part video series covering the entire discipline of biochemistry from start to finish in a comprehensive but high-yield manner. So without further ado, let's go ahead and get started on today's content. Today, I'd like to start off by talking about the urea cycle. The urea cycle is very high yield on these exams, so let's go ahead and break this down step by step. So the first step of the urea cycle mainly involves ammonia incorporation. The cycle initiates with the conversion of ammonia into carbamoyl phosphate, a reaction catalyzed by the enzyme carbamoyl phosphate synthetase 1, known as CPS1 in other diagrams. This step takes place in the mitochondria of liver cells and requires bicarbonate and ATP. Next, we have the formation of citrulline. Ornithine, a precursor derived from the breakdown of arginine in the urea cycle or from dietary sources, combines with carbamoyl phosphate to form citrulline. This reaction is facilitated by the enzyme ornithine transcarbamylase, also known as OTC. Sorry for butchering that pronunciation. Next, we have citrulline to arginine conversion. Citrulline is transported from the mitochondria to the cytoplasm, where it reacts with aspartate to form arganosuccinate. This reaction is catalyzed by two enzymes, arganosuccinate synthetase and arganosuccinate lyase. Next, we have arginine cleavage. Arganosuccinate is further processed by arganase, leading to the production of urea and regenerating ornithine. Ornithine can re-enter the cycle, ensuring its continuity. Urea elimination. Urea, the final product of the urea cycle, is water-soluble and less toxic than ammonia. It is transported through the bloodstream to the kidneys, where it is filtered and excreted in the urine. This process ensures the safe removal of excess nitrogen from the body. In summary, the urea cycle involves a series of enzymatic reactions that convert ammonia, a potentially toxic byproduct of protein metabolism, into urea. The cycle not only facilitates the elimination of nitrogenous waste, but also plays a crucial role in maintaining overall nitrogen balance in the body. Dysfunction in any of the enzymes involved in the urea cycle can lead to conditions like hyperammonemia, underscoring the importance of this metabolic pathway for human health. And I have some fun facts here for you guys to use for your notes. It's basically what I repeated verbatim to you guys in a more condensed manner. I'm not gonna go through and reread it. If you guys need to pause the video, take a screenshot for your notes, feel free to do so. I just mainly put this up here, so if anyone needs it for their notes, they can have it. Moving on, let's go ahead and answer some questions. So we have a neonate that presents with elevated serum BUN levels and normal creatinine. Further investigation reveals a defect in ammonia genesis. Which substrate is the most likely source of the elevated ammonia in this patient? All right, I think I gave you guys enough time. Let's go ahead and move on. If you need more time, pause the video. Moving on now. In cases of ammonia genesis defects where there is an inability to efficiently incorporate ammonia into organic molecules, the most likely substrate for elevated ammonia levels is glutamine. Glutamine is a key amino acid with a side chain containing an amino acid group, making it a significant source of ammonia in the body. The breakdown of glutamine releases ammonia, which is processed through the urea cycle to form urea, a less toxic compound. Therefore, in the context of impaired ammonia genesis, glutamine becomes a primary contributor to elevated ammonia levels. Therefore, the correct answer is C, glutamine. Let's go ahead and see why the other answer choices are incorrect. Alanine. While alanine does contain an amino group, it is not a major source of ammonia in the body. Glycine also contains an amino group, but is not a primary substrate for ammonia production in cases of ammonia genesis defects. Serine, similar to glycine, serine contains an amino group, but is not a significant contributor to ammonia elevation in this context. Valine is an essential amino acid that does not play a major role in ammonia genesis, making it an unlikely source of elevated ammonia in this scenario. All right, let's go ahead and move on to the next question. A neonate presents with elevated serum BUN levels and normal creatinine. Investigation reveals a defect in ammonia genesis. Which substrate is the most likely source of the elevated ammonia in this patient? Similar premise, so I'm just going to go ahead and move on right now. If you need more time, go ahead and pause the video. Moving on now. In cases of ammonia genesis defects where there is a failure to efficiently incorporate ammonia into organic molecules, aspartate becomes a key substrate for elevated ammonia levels. Aspartate is involved in the urea cycle, acting as a precursor for organosuccinate, a compound that contributes to ammonia elimination. Dysfunction in ammonia genesis can lead to impaired utilization of aspartate in the urea cycle, resulting in elevated ammonia levels in the neonate. Therefore, the correct answer is A, aspartate. Let's go ahead and move on and see why the other answer choices are incorrect. Leucine. While leucine is an essential amino acid, it is not a major source of ammonia in the context of ammonia genesis defects. Methionine. Methionine contains sulfur but is not a primary contributor to elevated ammonia levels in case of impaired ammonia genesis. Tyrosine. Tyrosine, an aromatic amino acid, is not a significant substrate for ammonia production in cases of ammonia genesis defects. Histidine. Although histidine contains an amino group, it is not a primary source of ammonia in the context of ammonia genesis defects. Let's go ahead and move on to the next question. A neonate exhibits elevated serum BUN levels and normal creatinine, indicative of a defect in ammonia genesis. Which substrate is the most likely source of elevated ammonia in this patient? Once again, super similar premise. I'm just going to go ahead and move on. If you need more time, pause the video. In the context of impaired ammonia genesis, arginine emerges as a likely substrate for elevated ammonia levels. Arginine participates in the urea cycle, where it is converted into ornithine and urea. Dysregulation in ammonia genesis may hinder the efficient utilization of arginine in the urea cycle, leading to the accumulation of ammonia. Thus, arginine becomes a crucial contributor to elevated ammonia in this neonate. Therefore, the correct answer is A, arginine. Let's go ahead and see why the other answer choices are incorrect. Proline. Proline is an amino acid involved in collagen synthesis and is not a major source of ammonia in the cases of impaired ammonia genesis. Cysteine. Cysteine contains sulfur but is not a primary substrate for ammonia production in the context of ammonia genesis defects. Lysine, an essential amino acid, is not a major contributor to ammonia elevation in the context of ammonia genesis defects. While threonine contains an amino group, it is not a significant substrate for ammonia production in cases of impaired ammonia genesis. So, what do you get from these questions? Basically, memorize the urea cycle pathway and understand what substrates can cause problems in ammonia genesis, because that's how I phrased those three questions. High yield, make sure you get it down. That'll be all for the video. I'll see if there's any more topics I would cover in biochemistry. If not, this should be the final part for the high yield review series. I might go into greater detail on certain topics that might pop up on the CBSE or the customized assessment service exams through NBME, so keep an eye out on that. If you have any questions, comments, or concerns, please feel free to leave them in the comment section below. If not, this is FixerMed signing off. Be sure to have a great day, everyone. Goodbye, and good luck studying.